So this is a presentation about the VisiSens A1 for oxygen imaging. And this is just a very short outline. So of course, just a short description, but we have already seen what the VisiSens is and what is to be used for. Some unique selling proposition, some technical data, and what I think it's really interesting because then you really see a lot of pictures and you can really think, oh, this customer is doing this, that maybe you can use the VisiSens to do this because a lot of other people have already done it. Components and pricing, and then again you have access to all kind of marketing materials and how to use videos, and then short summary. So, short description. So the VisiSens is a USB handheld microscope. Is it here? So well, you've already seen it, but it's really small, portable, simple to carry around. Then what it does, it visualizes the oxygen distributions as well as changes over time. And the time periods can be from seconds to months, like with our other oxygen sensor, so absolutely no problem. And the working range is between 0% and 100% air saturation. Yep. Next point, so basically, of course, you cannot just use the microscope, you also need to use our sensor. And it looks exactly like a sensor spot, but instead of being just a spot, it is a foil. So it is just a bigger membrane, basically. And the way you have to think about this is just, if you're on this foil, you have just thousands of microsensors, everyone really close to each other. So basically, every microsensor is giving the oxygen value, and then the software, what the software is doing is actually color coding. So for example, let's say that we want 0% air saturation to have a dark color, and 100% air saturation to have a bright color. Then of course when you color code your picture, you will see something that is basically that. So light color, lots of oxygen, dark colors, less oxygen, and you can take pictures in real time. So basically what you have at the end is just a slideshow or a movie that is very interesting for the customers, because they just take pictures every X seconds, minutes, hours, and then they can just have a very nice movie that show how the things is changing. And it differs somehow from the rest of our technology, because in this case you really get a very nice idea of where the oxygen is. Of course, microsensors are really precise, 30 micron size, you can just stick it in more or less whatever you want. But with this one you can even look at large areas, so centimeters, square centimeters, and then you really see where the oxygen is going. This is just a very nice pictures, because it just shows a laptop and a VisiSens, and that's everything that you need when you have to work with the VisiSens. So it's also portable. You can just go out in the fields, take with you your laptop, your VisiSens, and then you can measure for hours. So that's also quite nice. And well, this is just a very schematic view of how it works, but basically now that you're familiar with our technology, it's very similar. So you have, instead of having an optical fiber, you just have a camera unit. Then maybe you have some kind of wall, like a glass, and you just stick the sensor on the other side of inside the vessel, and then of course the sample is in contact with the foil. Yep. And well, of course, the VisiSens is coming with a software built from PreSens. The software is free of charge, and it allows you to control the image capturing process and also actually some image processing and evaluation. That is important, so that the customer can really measure quickly. You will see with the demonstration, it's very fast. And then also make some evaluation, so make some kind of statistical analysis and that kind of stuff. So just very short list of possible application: cell suspensions, tissues, complex access system, solution gels, and soil sediments and biofilms. But here what is interesting, so this is a microfluidic chip. So this is actually micro and nanotechnology. You just have some fluidic chip, and then the customer wanted to see how the oxygen was distributed inside this channel. So this is PDMS, the polymer. You can just, with some processes, you can basically shape it like with microchannels, mixing chambers, etc., etc. You just pour the liquids into one end, and then it goes. And these were just the regions that were recorded with the VisiSens. So you really see where the oxygen is more, where it's less, and how it's changing. And also this one is very nice. So this is just a plant. The customer wanted to have a look at rhizotrons. So basically just what he did was just put a sensor foil on the side, very close to the plant, looking at it, and then you can see the oxygen distribution inside the plant roots. And that's very neat. Sorry?
How do you read? Sensor?
In which sense? It's basically the same principle as with the optical fiber. So there is light that is coming out of the camera, and then of course the sensor foil is fluorescing. So light is coming back from the sensor foil to the camera. So basically inside you will see later, you have some LEDs for illumination, and then you have a CMOS sensor for capturing the picture. So, some unique selling proposition. Well, what is interesting, it is not invasive, like our rest of our technology. It's very flexible because it's USB powered. For example, I really love this picture. This was made in Spain. There were some researchers that actually wanted to investigate. Basically, it was a very old bridge made by the Roman, and the bridge was being corroded by aggressive mushrooms, and they wanted to test different kinds of pesticides, and to check how the pesticides were affecting the mushrooms, they just took the oxygen values with a VisiSens. So really just, I liked it because it's really, you can work outside, in the light, just need a laptop, maybe an extra battery, and then the VisiSens, and everything is perfect. Then this is somehow what I tried to explain before. So you have one device instead of having thousands of individual sensors, because basically every pixel that you see on the camera has to be considered as one oxygen microsensors. Then, well, okay, once again, you only need a notebook or a PC, and there is no additional power source needed. So you see it in my hand, it's just one cable, the camera, everything is there. And then of course you can also take imaging of living samples. So like in the third picture, you can actually see there is the example from before. So the plant is growing inside the vessel, there is a sensor foil in there, and you just come with your VisiSens on the side, and you'll have a look at how the oxygen is changing. So technical data, this is not really important, but so it's two-dimensional optical sensing, USB handheld microscope. It's very simple. So you have a CMOS chip, some lens for magnification purposes, and optics blocks, so 8 LEDs for illumination, for the excitation. Then of course you have some filters to filter the light that is going back to the detector, but that's it. And well, we've already seen this light today, so it basically...
That is because I skipped it a bit rather fast. For me that is very important that you perhaps just once in your life understand how we make our sensors. So we take an indicator which is the fluorescent dye, something that is re-glowing. Give light, get back. Then polymer is not completely correct. Most of the time it's a monomer that later on gets to a polymer and some solvent. Then we make a sensor cocktail just by mixing. And then we have different possibilities. Typically we make, either we give the cocktail direct, for instance, on the microsensors, on the tip of the glass fiber, microsensors ready. Or we just make, and that is the second thing, we make a layer just by cut knifing it, and afterwards by drying it, for instance with UV light or with temperature, something like this. And then we got foils that really are hard like this, polymer, completely pink color, for instance, and then the ladies came and stamp out the sensor spots. And if we do not stamp out the sensor spots, but just take a foil, a 4 by 4 centimeter foil, we got the things for the VisiSens. Basically, this is rather simple, but you have to see it once. So these spots are typically stamped out. Sorry.
Yeah, no problem. So, this is how the sensing process is working. And it's really similar to what we've seen before. So there is some excitation light going from the camera to the sensor foil, and then of course there is some emission from the sensor foil to the camera. What is important is that of course if you have something like a vessel, you actually have to put the sensor foil inside the vessel when it's in contact with the thing that you want to measure. So with the solution, with the gas, with your plant, with your roots, whatever. And well, of course, you can put the sensor into a vessel. You can also directly measure. For example, on your skin, we will do that kind of experiment later. So you just directly cover the surface that you want to measure with the sensor, and then you measure. Or, of course, you can just place it inside some kind of vessel, like some transparent window, or it really depends what you want to do. So a lot of people, for example, that are working with plants, they just use a transparent glass container vase, stick the sensor on the side, they hold it, they tilt it a little bit, so that the plant roots are really growing on top of the sensor, because it has to be in close contact what you want to measure. So of course you will not get the complete picture if your vessel is 2 meters long. I mean, you will just see the oxygen distribution that is in contact with the sensor. That is important. Sometimes people just try it and they just glue the sensor 10 centimeters away, and there is soil in between the roots and the sensor. In that case, what you see is the oxygen in the soil, not really the oxygen in contact with the roots. This is quite technical as well, but I have to explain this. So, of course, it's a microscope, so it has a resolution, a field, and a field of view. So, the field of view is actually even given by the distance that in between the detector and the sample. So, we have these so-called adapter tubes. They are useful for two purposes. So, the first one is actually to avoid any incoming light, stray light from the outside. Because in this case, the light that is coming from the VisiSens is not frequency modulated. So we are not measuring the phase in this case. We are measuring the intensity. So stray light...
The intensity?
Yeah.
Of red and green.
Yeah, of course there is some filters.
Different beams, which makes it nearly bulletproof also. So not really the intensity, but the intensity of two colors. And while we're using the quote in between red light and green light, everything is divided. It nearly somehow get rid of the mistakes with the kind of technology with it. You don't have to know it, but it's not phase, it's just red and green.
So basically, of course, the working distance, if you place the end of the adapter tube just in close contact with the foil, it would give in the field of view and the resolution. And we have three different kind of adapter tubes, but basically the one that is sold is always 99.9% of the cases just the so-called adapter tube 1. So in this case, the pixel resolution is about 10 microns, and the field of view is about 1 square centimeter. Then of course you can have, well, larger adapter tubes, longer adapter tubes, and you have larger field of view but worse resolution. Or a shorter adapter tube, and then you have much better resolution, but the field of view is smaller. I will show it also practically later, but just so that you know that you have different adapter tubes. But as I said, 99.9% of the cases, the customer needs this one. So some specification of PC unit requirements. So unfortunately, it doesn't work on a Mac, only on PC. But Windows XP, Vista 7, and I think that Thomas also did some tests on Windows 8. So it was basically working.
Does anybody have Windows 8? Are you happy with it?
Yeah.
It nearly killed me. So you've got no start button, but in version 8.1, and the thing with the Metro apps nearly killed me. It took maybe a quarter of a year to get used to it.
The latest update we can have the same similar Windows 7.
I've got the Windows start button now.
Yeah.
Due to some freeware.
I read the one, I think the one.
Windows 8.1.
You just update it. You can get the Windows 7 abilities or functions.
On private computers, I just took a step back and got to Windows 7 again. Sorry.
Yep. So technical data, just the most important. So the measuring range is between 0 and 100% air saturation. Field of view is 1 square centimeter with the normal adapter tube, and the resolution is about 10 microns. That's everything that you need to know. Software. So the software is coming for free with the VisiSens. It's already included in the sales package. and it's quite easy to use. So basically you just take pictures, and then you have the so-called evaluation mode. When you took the picture, so for example this one, red and green, here you can see the ratio between the green and red light coming from the sensor, and then you do some processing, and then what you see is actually this one. Then in the evaluation mode you can also have a look at cross sections, at the evolution of the oxygen in a certain area over time, histograms, and other kind of information that are useful for the customers, for the user. And that's the part of the presentation that I think it's really interesting. So the application fields. So the VisiSens, it really has a lot of different application fields, basically hundreds. It's a relatively new product. It's been on the market for a bit more than 1 year, but we have been looking at really a lot of scientific publications. So I think that the last list that I saw was a couple of months ago, and basically in less than 1 year, people managed to write at least 60 publications in good, high impact factor journals. So it really is a very nice scientific tool. So of course, well, kind of the obvious application is in plant and root systems. We already seen some pictures, so for life science, I would say it's very nice. This is a microfluidic chip. They integrated the sensor foil inside the chip, and then this just makes some flows inside. There is a mixing chamber, different stuff is coming out from the different sides. Then this is very good for biologists, sediments, limnology, everything that has to do with the study of sand, soil, water interactions. So soil, sediment, biofilm. This is just a tank filled with sand, some water, and they look at actually how the oxygen is changing when they change the conditions. Also, why not cell culture? So this is just some dishes for cultivation. They put some, just cut the sensor foil and put inside the dishes. Just have a look with the VisiSens how the cell culture is going. So you really see not only just one oxygen point, but also the distribution inside the dishes. Animal-human physiology, well, actually only animal. It's not a good idea to, well, to cut someone open and put a VisiSensor foil.
Especially it's forbidden, so
yeah.
PreSens meters are not for human applications.
Exactly. So I will show you a very nice demonstration that you can do with your skin, but of course it's on the outside. I don't want to cut myself or something like that. This is also quite interesting, so process control, material research. Actually this is talking about wine. This is a wine barrel. So they just made this kind of transparent window into oak barrels, and then they're just looking at the oxygen distribution inside for the wine, because of course it's an important parameter to watch out. This is what I was telling before, so this bridge in Spain that was being corroded by mushrooms. So just studying how the plants were reacting to the pesticides. Packaging, why not? The same principle as before. So you don't get just one point, but a distribution. And then of course some special solution for engineering OEM. And what is also nice here, we have a link, a direct link to our web page. So if you just click here, then you can just go on the web page and you will find all kind of description, application notes. Where actually, I mean, if you just go to a customer that is doing plant research, you just print it and go to him and everything will be clear. So, some nice example, plant physiology, a leaf of a living plant. So actually the leaf is still connected to the plant and the plant is living. This is necessary because the respiration in plants takes actually a lot of time. So, well, if you go to a customer and he wants to measure a plant, and a lot of customers do, if you want to do that, then you have to stay with the customer for at least 6 hours. So I will not advise to do that, but it's really nice to show them this kind of pictures. So just very high magnifications, and really how the oxygen is looking inside the leaf. So you can really have a nice resolution and just appreciate where the oxygen is.
So two things that Daniele said is very important. First, never make a demonstration with a leaf. Never ever. 6 hours is a minimum.
Minimum, but it can be much longer.
Sometimes you have to wait for 2 or 3 days. That's absolutely ridiculous. What Daniele is showing is a 3-minute experiment.
Yeah.
First, second, there are lots of guys out there, even if they are coming from biology, they just cut the leaf and just give it onto a plate
and then of course it's completely dead.
It's not working. You know, most of the leaves are instantly dead. After they are cut, it's like cutting your head. So you have to tell them that they are still living and watered and everything like this, and still are connected to the rest of the plant, including the rhizostomes or the root system.
Yeah, exactly. But it takes longer, unfortunately. So
basically,
that's why plants are not running around.
Like a dead fish that they would try to I don't know, get into life or something. It's ridiculous. But you have to tell them sometimes. Right?
Yeah. So we also made some measurement in oxygen distribution in seeds. So of course, inside a lot of seeds there are some antioxidants to prevent the oxygen from going inside. And we just cut the peas in the middle, two different kind of peas. And then you can actually look how the distribution is changing depending of the, well, basically on the structure of the seed and on the material composition on what's inside. And the graphs that you see are quite nice. These are just the cross section that you see here, the dotted line. So basically everything is made with the evaluation software. So if you did give this kind of information to a scientist, I think he would be quite happy. It's for a publication. I mean, it looks really good.
So this means the antioxidants stuff is in the middle. It means there is no oxygen in the middle of that kind of embryo. So you know that vitamin C is called an antioxidant. Antioxidants means anti-oxygen, means reducing any kind of oxygen, and of course also of aging things, and that means the blue indicates a lack of oxygen.
Yep. Hopla. This is another nice experiment. They took a membrane and they made just a fouling simulator. So basically they let it rot. So these are oxygen pictures, and here is just a normal picture on the other side of the membrane. So you can see that basically at the beginning the membrane is quite clear, nice, and then there is just all kind of dirt, bacteria, stuff that is growing on it. And of course some of the bacteria actually are consuming oxygen, so the oxygen was going down. So also this is quite a nice example, a complete different field with respect to what we have seen before, but it's still quite nice.
Is biofilm known? The definition of a biofilm, what it is? It's not. No, it's not coming from biotechnology. If you're going to the sea or to a lake, you just stand on the stone, and sometimes they are very slippery. And that is a biofilm. Basically, this is made a layer of bacteria, something like this. Bacteria, dirt, and all that kind of stuff, who typically is also a bit growing and makes lots of problems in the laboratory, where you just have to use some very toxic things to get rid of biofilm. For instance, with the clock type electrode, that one is can be attacked by biofilm typically. So it's a slippery film on the top of the membrane.
Another example is what seen before. It's basically the determination of oxygen gradients in engineered tissue. So there is just a sensor foil inside one dish, and then you just look and you just monitor it. But in this case there is, well, just to see how it can be placed to look at this kind of vessels. Perfusion monitoring in skin, so basically to see how the oxygen is changing with time inside your skin. And it's very nice, but because as I said, basically then you have a time series of pictures and you just have some kind of slideshow or movie. It's very clear. This is not a human brain, luckily. It is a mouse brain. So what they did, they put the sensor foil on top of a PDMS, just a transparent cylinder, put inside adapter tubes, and then just looking at how the oxygen was changing. Unfortunately, well, for the mouse, not a very nice experiment, but it's a possible application. So people have been doing that successfully. And you can see that how in the brain, with the time, the oxygen is just flowing from some region to the other ones.
Do you have some animal physiologists in your customer base? They sometimes have strange experiments.
Yeah.
Especially with microsensors in the eye of fishes, of course, that they were not conscious, fortunately. In the brains of rats, in the brains of mice, in the back of butterflies. So it's all a bit Frankenstein experiments. But they love that kind of pictures. Oh, great.
Yeah.
So, a bit weird, but does not help.
Yep.